Dear students, welcome back. So in this session, what will I be talking to you about? I'll be talking to you about another important pathway, hexose monophosphate shunt, HMP shunt. Also, which are the other names? Hexose monophosphate shunt, HMP shunt, pentose phosphate pathway. It is also called as pentose phosphate pathway and phosphogluconate pathway. These are the different names of HMP shunt. Again, entrance point of view, you will have to know it, the different alternative names. Now, will I be telling you the pathway? Again, as I have told to you, I don't believe in telling you our reactions. What are the reactions that are taking place, the whole pathway? Why? Those are usually asked only in short answer questions. And here we are looking at MCQs. So with this focus in mind, I'll be talking to you about the importance of each of these pathways. And there is enough importance of these pathways to know about, even without knowing what reactions are taking place. So let us forget the pathways. Let us focus on the importance of these different pathways. Now, HMP shunt. What is it? It is an alternative route for oxidation of glucose. So again, is it a catabolic pathway? Yes, here also there is oxidation of glucose that is taking place. Hence, it is a catabolic pathway. Now, does it generate energy? Now, as I said, if you say catabolic, that means it has to give energy. But no, this does not generate ATP. The most important thing about HMP shunt, MCQ point of view, every point of view, HMP shunt does not generate ATP. So, does it take place in RBCs? Yes, it does take place in RBCs. RBCs also use glucose by this alternative pathway, but here purpose is different. I'll be talking to you about it. So each pathway has its own purpose. Now the main thing of purpose of HMP shunt is to produce NADPH and pentoses. Do not forget the difference between NADH and NADPH. Okay, the main important function of HMP shunt, so it produces NADPH. NADPH is produced. The reduced form of NADP is NADPH that is produced, and it also produces pentoses or pentose phosphates are produced in HMP shunt. That is the main purpose of this pathway, not to generate energy but to produce NADPH. I'll be talking to you about the uses of NADPH also in this session. Now, carbon dioxide is also produced here, so that's why it is oxidation of glucose. Nearly 20% of glucose is oxidized by this pathway. Where is it happening? In most of the cells, in all the cells, HMP shunt takes place. It is not restricted to only one tissue. It happens in all the tissues. Wherever we have the need for pentose phosphate, HMP shunt takes place. Remember, glycolysis also takes place in all the tissues, and it takes place in the cytosol. Similarly, HMP shunt also takes place in all the tissues and in the cytosol itself. HMP shunt is also restricted to the cytosol of all cells. Now, where is it more? Where there is maximum HMP shunt taking place? Now, these are the tissues which have a greater need for NADPH. Look at the purpose of the pathway, to produce NADPH. So, wherever NADPH is required, that is where this HMP shunt is happening at the maximal level. So which are they? They are the tissues which are involved in reductive biosynthesis. I'll talk to you about which are these tissues and what do you mean by reductive biosynthesis. Please remember, NADPH is more formed via the HMP shunt in tissues which are involved in reductive biosynthesis. So which are these tissues? Here I have a list. Mainly in the RBC. Why? To maintain the reduced glutathione. I'll talk about what is glutathione as we go ahead. It also occurs in the WBCs. Why? Generation of superoxide and helps in the phagocytosis. It happens in the adipose tissue. Why? Again, reductive biosynthesis, anabolic pathway. Fatty acids are synthesized in the adipose tissue, and triacylglycerol is synthesized in the fatty acid in the adipose tissue. So, hence, fatty acid synthesis has to take place in adipose tissue, and hence we require HMP shunt is very active in adipose tissue. Mammary gland. Yes, again, HMP shunt is required because in the lactating mammary gland for the production of milk. Milk contains lot of fatty acids, and these fatty acid synthesis, again it is a part of reductive biosynthesis, and HMP shunt is required. It also very active in the hepatocytes. Please remember that most of the pathways are active in the liver. Most of the pathways except ketolysis. Other than ketolysis, all other pathways happen in the liver tissue, hepatocytes. Okay, so in the liver it has different role. It can be used for fatty acid synthesis, cholesterol synthesis, bile acid synthesis, etc. In the ovaries, in the testis, in the adrenal gland, again HMP shunt is very active. Why? These are the tissues where steroid hormones are synthesized, and for steroid hormone synthesis, please look here, everywhere I am talking of synthesis. Reductive biosynthesis requires NADPH, and HMP shunt produces the NADPH. So, what are the reactions of HMP shunt? Again, I am just telling you the names of the enzymes and not the pathway. It has two phases, oxidative phase and the non-oxidative phase. The oxidative phase is irreversible. It does not change. You cannot bring it back, to glucose. The oxidative phase is irreversible. The non-oxidative phase is reversible. There are so many reactions that are taking place which are keeping on reversing. Reversible reactions are taking place. So depending upon what the body needs, the non-oxidative phase will produce that. I will be talking to you about each of these. So the non-oxidative and oxidative phase is irreversible. Now in this, please remember that glucose is already have to formed glucose 6-phosphate. Could be glucokinase or hexokinase. Now comes the role of glucose 6-phosphate. Now this is one reaction which I am going to talk to you about, which I am going to illustrate. Please remember, glucose 6-phosphate undergoes what? It undergoes dehydrogenation. By what? Glucose 6-phosphate dehydrogenase enzyme. And again, this is an important enzyme because its deficiency can lead to so many diseases. And in this process, please remember, glucose 6-phosphate dehydrogenase, the coenzyme is NADP. And it is a irreversible reaction. And NADPH is produced. NADP is used and NADPH plus is produced. So NADPH is produced, it gets converted to phosphogluconolactone, and intermediate and all is formed in that. But most importantly, it forms phosphogluconate. I'll just tell you phosphogluconate. The intermediate is not so important, but more important is the next enzyme. The next enzyme is again 6-phosphogluconate dehydrogenase. Again a dehydrogenase in which again NADPH is produced. These are again 6-phosphogluconate dehydrogenase and glucose 6-phosphate dehydrogenase are the two dehydrogenases in the body where NADPH is produced. That's the importance of the oxidative phase. Glucose 6-phosphate to 6-phosphogluconate, and further it goes on. As I said, I am not focusing on the reactions. What is important here is NADPH is produced. Now what happens? Then starts the non-oxidative reversible phase of HMP shunt. Now there is one more enzymes. I have just given you the list of enzymes. Ribulose 5-phosphate epimerase, ribulose 5-phosphate ketoisomerase, transketolase, transaldolase, transketolase. So these are the 5 enzymes which are involved in the reversible phase. Please note only the names of the enzymes. Ribulose 5-phosphate epimerase, ribulose 5-phosphate ketoisomerase, transketolase, transaldolase. What is important here is the enzyme transketolase. What does this enzyme mean to you? Transketolase has a requirement for a coenzyme. The coenzyme is thiamine pyrophosphate. TPP is the coenzyme for thiamine pyrophosphate. TPP, that is thiamine pyrophosphate, is the coenzyme for transketolase. So in TPP deficiency, that is in thiamine deficiency, we estimate the levels of erythrocyte transketolase. So it helps us to know whether TPP is enough or not, whether erythrocyte transketolase activity is good or not. We come to know by measuring the activity of transketolase, and helps us to know about thiamine deficiency. So this is the importance of the HMP shunt. Now these are the different enzymes which are involved in the reversible phase. So what are the uses of HMP shunt? As I've already told you, one most important is using, it helps in the production of NADPH. What else is the use? The second use is, during all this reaction, ribulose 5-phosphate is formed, which will be again converted to ribose 5-phosphate. So what is ribose? Ribose is a pentose. What is ribose 5-phosphate? What is ribose 5-phosphate? It acts as the precursor for the de novo synthesis of nucleotides. Where are nucleotides seen? In the DNA, in the RNA. So cells which require pentoses will produce pentoses by the HMP shunt pathway. So NADPH is produced in the oxidative phase, and it happens in tissues which have a greater need for NADPH, as I said, where reductive biosynthesis is occurring. Whereas ribose 5-phosphate, which is the pentose that is formed, is required for nucleotide synthesis. So the non-oxidative phase occurs in most of the tissues. Wherever DNA synthesis or RNA synthesis is taking place, de novo synthesis of nucleotides is taking place, ribose 5-phosphate is produced by the HMP shunt. Now what is happening here is, the non-oxidative phase is made up of reversible reactions. Hence the tissue decides what to do. Should I produce NADPH or should I produce ribose 5-phosphate? If ribose 5-phosphate goes off, the amount of NADPH produced will decrease. Why? Otherwise fructose 6-phosphate and the intermediates are formed, which will come back into the HMP shunt and produce more NADPH. So depending on the tissue need, one of these will be produced. So what are the uses of NADPH? I kept telling HMP shunt produces NADPH. What are the uses? Is it used for ATP production? No. Remember, NADPH does not enter the oxidative phosphorylation. NADPH will not produce ATPs. It is only NADH which produces ATPs. So that is the difference which you cannot forget. It is used for reductive biosynthesis, oxidation reduction reactions, for detoxification. It is used for phagocytosis. It is used in the synthesis of nitric oxide. So we'll look at all the different uses of NADPH one by one. Reductive biosynthesis. Which are the reductive biosynthesis? Fatty acid synthesis, fatty chain elongation, cholesterol synthesis, steroid hormone synthesis, neurotransmitter synthesis, deoxyribonucleotide synthesis from ribonucleotide. For all this, we use NADPH. Look at the different reactions which use NADPH, and this is restricted only to the reductive biosynthesis. Okay, now we go ahead with the other use of NADPH in oxidation reduction reactions. Where have we heard of this? Oxidoreductases. Yes, it is used in the reduction of hydrogen peroxide. Hydrogen peroxide is commonly produced very much, and it has to be very toxic to the cells, and it has to be reduced. And for the reduction of hydrogen peroxide, we use NADPH. NADPH is also used for detoxification of drugs. There are so many drugs which need to be detoxified. All drugs that we take are toxic to the body. Many drugs, most of the drugs. So hence they need to be detoxified. And this is taking place in the liver. And in the liver, this is done by an enzyme called as monooxygenase. And this requires NADPH. Now, if you look at it, it is the cytochrome P450 monooxygenase which requires NADPH. Now, what is the cytochrome P450? We keep talking about them. Now, cytochrome P450's enzyme systems are a super family, not one enzyme, but a big family, a super family consisting of many heme containing enzymes, lot lots of enzymes are there, not one or two enzymes. We talk of a super family. All the enzymes of that contain heme, and they participate in a variety of reactions, including detoxification of drugs, detoxification of xenobiotics, detoxification of steroids, alcohol. In all these cases, the cytochrome P450's enzyme systems are involved. They are the ones which are responsible for various drug interactions also. Certain drugs will cause induction of the cytochrome P450 enzyme system and lead to higher metabolism of other drugs. So hence it is important to know about cytochrome P450 monooxygenases, and they require NADPH. Now the other uses of NADPH, one important use, as I said, is in phagocytosis. Reductive biosynthesis is over, oxidation reduction reactions are over. Now we come to phagocytosis. What is phagocytosis? Phagocytosis is the process by which WBCs engulf a bacteria and they kill that bacteria. Okay, simple way to tell it is they kill the bacteria. How do they do that? How does a WBC do that? So bacteria which are opsonized, we call it the term opsonization. So bacteria which are opsonized are engulfed by the WBCs. It is a type of endocytosis in which a bacteria, I'm just going to show you bacteria. So this is the bacteria which has been opsonized. It's a crude diagram to show it. So when this is opsonized, this bacteria will be taken up upon by a WBC. So a WBC will phagocytosize it. So when it phagocytosizes it, what happens? It will form a phagosome. So it will go and engulf this. So it will engulf this. You know the process in this. So it will engulf this and it will form a phagosome in which the bacteria is present. Now this phagosome, what happens in this WBC? Further what happens? This phagosome will combine with a lysosome. The lysosome, remember the lysosomes are a subcellular organelles. These lysosomes have pH less than 5 and they have lot of enzymes which can degrade bacteria. So it will form a phagolysosome. This is a phagolysosome in which the lysosomal enzymes have come and there is a bacteria with its opsonized product or whatever. Now this enzyme has to degrade this bacteria. It has to degrade this bacteria. So how does it do that? These enzymes in this phagocytosis, there is an important, very important enzyme which is present, and that is NADPH oxidase. So this phagocytosis, it is a very important defense mechanism in bacterial infection. So I have shown you here the phagolysosome. So in this, oxygen is utilized and it is converted to the superoxide anion by NADPH oxidase. This O2 dot superoxide anion is during this process there is a lot of use of oxygen by the phagolysosome or by the WBC, and this is called as the respiratory burst. So when a respiratory burst occurs, lot of oxygen is used and oxygen is converted into the superoxide form. Superoxide anion is further converted to hydrogen peroxide. So this superoxide anion and hydrogen peroxide, both of them are very toxic and they will kill that bacteria. They will bring about bacteriolysis in the phagolysosome. So in this, what has happened? For the production of superoxide anion and hydrogen peroxide, we require NADPH. So from where did NADPH come? From HMP shunt. So NADPH oxidase is another very important enzyme that is present in phagocytosis. There are conditions in which NADPH oxidase is deficient. I will talk about it later. So right now the focus is on NADPH. Now another important use of NADPH is in the synthesis of nitric oxide. Have you heard of nitric oxide? If you have not, It is time you hear about it. It is the molecule of the century. The 21st century molecule is considered to be nitric oxide because it has so many uses, so many things, so many drugs act via nitric oxide. Everybody should know about nitric oxide. So how is it synthesized within the body? And what is the role of NADPH in its synthesis? Remember, nitric oxide is written as NO. Nitric oxide with a superscript dot in it. It is a free radical that is produced, NO dot. So how is it synthesized? It is synthesized from arginine. What happens? Arginine, what is arginine? Arginine is a amino acid. What happens to arginine? This arginine is converted to citrulline. Have you heard of citrulline? Citrulline is a metabolite involved even in urea cycle. So arginine is converted to citrulline, and during this process NADPH is converted to NADP+. But along with that, oxygen is also used and nitric oxide is formed. What's the name of the enzyme? Nitric oxide synthase, NOS. The name of the enzyme is NOS, nitric oxide synthase. There are different isoenzymes of NOS, neuronal NOS, inducible NOS, all these different types I'll be talking later. Right now let us focus on HMP shunt, and that NADPH is required for the synthesis of nitric oxide. So what is the role of NADPH in RBCs, where I said it is very active? Please remember that it is to maintain glutathione in the reduced state. This is essential to preserve the integrity of the RBC membrane. Again, I have told to you, RBC membrane is very fragile and it needs to be maintained. And in RBC, the only source of NADPH is the HMP shunt. And this also keeps the iron of the hemoglobin in the ferrous state and prevents the formation of methemoglobin. Is there any deficiency? As I said, there is one important deficiency, glucose 6-phosphate dehydrogenase deficiency. It affects more than 200 million individuals. It results in the cause of hemolytic anemia. It is X-linked. They have inability to detoxify oxidizing agent. Is it an evolutionary advantage? Supposed to be. Increased resistance to falciparum malaria is seen by the female carriers of this mutation, and hence it is supposed to give an evolutionary advantage. How does it cause hemolytic anemia? So whenever there is decreased NADPH, it will mean that there is decreased glutathione. If there is decreased glutathione, there is no detoxification of hydrogen peroxide. So what? It causes oxidation of the membrane proteins. Glutathione also leads to methemoglobin, which again leads to hemolytic anemia. When there is oxidation of membrane proteins, RBCs become rigid and non-deformable, and this also leads to hemolytic anemia. Glutathione causes sulfhydryl groups of proteins get oxidized, denature proteins including hemoglobin, and they will lead to Heinz body formation, and these are attached to the RBC membrane. So this is a mechanism of hemolytic anemia that is seen in G6PD deficiency. Are there any precipitating factors? Yes, they develop hemolytic anemia when treated with oxidant drugs like antibiotics, anti-malarial, antipyretics. We call them the 3 A's drugs which precipitate G6PD hemolytic anemia. Favism also precipitates an infection where there is generation of free radicals in macrophages which diffuse into the RBCs and cause oxidative damage. So that also leads to hemolytic anemia. So with this we come to the end of this session on the HMP shunt, uses of HMP shunt, and the disease which is associated with the enzyme deficiency of HMP shunt, that is glucose 6-phosphate dehydrogenase. Thank you.